Good day and welcome once again to our YouTube channel. Thanks for subscribing to our channel, we really appreciate it. Today we quickly want to have a rundown of the specimens that will be used for the biology practical of the WAEC 2023. WAEC 2023 biology practical specimen. Those are the things that we want to look at today. So first, before we go deeply into explaining the characteristics or some of the possible questions that will be seen in the specimens, we'll quickly have a listing of the specimens. So we're starting with Specimen A. Specimen A Specimen A is said the mature fresh eggs of catfish. Mature fresh eggs of catfish. That is our Specimen A. Our Specimen B is fresh egg of domestic fowl. Fresh egg of domestic fowl. He said it should be raw and with its shell intact. With shell intact. C, Specimen C, will be having picture, model or chart showing the uterus containing fetus. Picture or model showing the uterus containing fetus. That is our Specimen C. And our Specimen D. Our Specimen D is the longitudinal section of ovary of Pride of Barbados. The longitudinal section of Pride of Barbados flower. That is our Specimen D. And our Specimen E, they said the leaf of Pride of Barbados. Leaf of Pride of Barbados. And for our Specimen F, our Specimen F, we shall be having the Panicum plant. The Panicum plant. That is what we will be having for our Specimen F. Panicum plant, popularly known as the Guinea grass. Popularly known or called the Guinea grass. That is our Specimen F. So we'll be needing the whole plant, the whole Panicum plant or the Guinea grass for our Specimen F. And our Specimen G is the Cocoyam plant. Specimen G, Cocoyam plant. Cocoyam plant. Botanical name is the Caladium plant. The Caladium or Caladium plant. That is what it is called. We also be needing the whole plant for that. And our Specimen H. Our Specimen H is the comb of Cocoyam. The comb of Cocoyam is going to be our Specimen H. And our Specimen J now, Specimen J is going to be the dry humus in beaker. We're having dry humus. And for our Specimen K, we'll be having wet or moist humus. Wet or moist humus, that is what we'll be having as our Specimen K. And our Specimen L is going to be ripe orange fruits. Ripe orange fruit. That is what we'll be using for our Specimen L. And our Specimen M is going to be longitudinal section of coconut fruit. The longitudinal section of coconut fruit. That is going to be our specimen M, and specimen N, which is going to be our last specimen for this particular experiment. Our specimen A, or practical. Our specimen N is going to be the longitudinal section of fresh chili pepper, fruits. So these are the specimens that we will be having for our practical as we move on in the experiment. So moving further now, we'll be picking each of these specimens one after the other. We'll be starting from our specimen A. So that is what we'll be having now. We want to pick all of our specimens starting with the specimen A. Specimen A. We know that our specimen A is the mature fresh egg of catfish. Specimen A, mature fresh eggs of catfish. So that is where we want to start from. Specimen A is the mature fresh egg of catfish. So we are going to have to explain this quickly. Now, when we look at the fresh egg of catfish, okay, let me start by telling us the botanical name of catfish. Catfish is called, we have it called the Clarias garipinus. Clarias garipinus. That is what we refer to the catfish as, Clarias garipinus. So we need to have that at the back of our minds. Now, let me quickly show us what the egg of a catfish looks like. Looking at this particular diagram, this is what the egg of a catfish looks like. We can see it. We can see the eggs inside a particular envelope, kind of a sheet, kind of, and that sheet has all of the tiny cells in it, the tiny cells that look pale yellow in color. We can see all of these things here. We can see them. They look pale yellow in color. So those are the cells that make up the egg cell of the catfish. Are we together? So the catfishes produce or lay large number or large volume of eggs. They produce or lay large volume of eggs, and these eggs have numerous cells with diameters ranging from 2.4 to about 3 millimeters. The diameter of each of the cells of the eggs have 2.4 to 3 millimeters. That is the diameter, and the eggs are usually transparent right before they are laid. The eggs are usually transparent right before they are laid, and by the time they get laid and a couple of minutes after being laid, you find out that the eggs are already changing color to let's say pale yellow to about orange in color after a couple of minutes when they are laid. And if we look at the temperature of the particular temperature for hatching the eggs, the conditions necessary for hatching the eggs, you find out that the temperature usually needs to be about 21 to 29 degrees Celsius. The conditions, we have the temperature for the hatching the eggs usually going to be between 21 to 29 degrees Celsius. And these eggs, they hatch about 5 to 10 days after they are being laid. So if we look at this particular picture here, we have the catfish having the eggs inside it. This is a mature catfish having the eggs inside it. So that is what the egg of the catfishes look like. So we quickly said, let's quickly have a summary of what we have said. We said that the catfishes, they lay large volume of eggs. Catfishes lay large volume of eggs. Large volume of eggs. And that their eggs have about, we have each the cells of the egg have between 2.4 to 3 millimeter in diameter. They are 2.4 to 3 millimeter in diameter, and usually they are transparent before being laid, changing to we say pale yellow or orange, after being laid. And the conditions necessary. One is that the temperature has to be between 21 to 29 degrees Celsius, and two, that the hatching period is between 5 to 10 days. After the eggs are being laid. And because the catfishes, they lay eggs, we refer to them as oviparous organisms. So you could be asked that as well. So that is what we have for the specimen A. Let's go to specimen B now. Specimen B. We have specimen B. Specimen B is the fresh egg of domestic fowl. That is the specimen B. So for the specimen B now, we say that the eggs have cancerous nature. They have cancerous nature because they give or they produce mechanical support for the contents. The shell of the egg, like we can see here from this very picture. We can see that the egg, they have a shell. Now, oftentimes the eggs come in two major colors. The shell of the egg come in two major colors. It could be brown, like we can see in this particular one here. We can see that all of the eggs here, they are the ones that I'm pointing at, they are brown in color. And also we can have the shades of white. We can have them to have what? White color. So those are the two major colors that we have for the domestic fowl, for the eggs of the domestic fowls. Now, like I said earlier on, we could have the eggs have their shell to contain they are cancerous in nature, and this makes it possible for the eggshell to provide mechanical support or mechanical protection to the contents of the egg, to the internal structure of the egg. Also, the shell is porous. As such, it is able to give gaseous exchange with the environment. Now, at this point, we need to understand that the shell houses or contains the embryo of the organism. It contains the embryo of the organism. So from this very picture here, we will be able to see that the egg here, the outer shell, has a content that it is protecting. The content of the egg, we have the outer shell, we have the calaze, we have the germinal disc or the blastoderm, we have the yolk. This particular little part here that is painted red in color is the germinal disc or the blastoderm, and then we have the yolk that is rich in protein. So also is the albumin. The albumin, this white part here, is called the albumin. This part here, it is not labeled in this particular diagram. I could have it labeled as such. We could have it labeled as such, something like this, having this as the calaze now. As the albumin. could have it labeled as the albumin. Are we together? So as you can see, like as it has been labeled, we have this part here called the albumin. So the albumin and the yolk, they are very, very rich in protein substances. So like I said earlier on, that the eggshell has calcareous nature. It gives mechanical protection to the content, and then the eggshell is porous, so allowing for possibility of gaseous exchange with the surrounding. And then the shell houses or contains the embryo. Inside here, it is this yolk that eventually forms the embryo. It contains the embryo. The shell is brown or sometimes white in color. Like I have said earlier on, that the shell could be white or it could be brown in color. Now, the albumin is a clear liquid in the raw egg. This part here, the albumin, is usually clear. It's usually a fluid liquid. It's a liquid that is clear in color, but it turns white after after the egg has been allowed or made to pass through heat. So when you boil it, the liquid transparent color changes its color to the white coloration. And then we have the yolk also changing to yellow. Now, the white part of the egg has about 67% of the protein substance in the egg itself. So unlike like the statement you probably might have heard that the yolk is very important. Yes, the yolk is very important, but even the albumin has more protein content than the yolk. The protein content in the albumin is about 67% of the total yolk, of the total egg percentage. So we have that for the eggs. And then we said the yolk contains carbohydrates and generates energy for the embryo. The color of the yolk deepens to yellow at the center after heating, and then it hatches after 21 days. The egg hatches after 21 days. So that is what we have for the egg. So let us quickly list a couple of characteristics for it. We said the eggshell. Excuse me. We said the eggshell has calcareous nature, giving mechanical support or protection to internal structures. We also said that the shell is porous. For gaseous exchange. We also said that the egg, we have said that the egg could be white, the shell could be white or brown in color. We said that earlier on. Also, that the albumin and the yolk are highly rich in protein. So these are few of the characteristics that we'll be talking about for the egg. Also, that the gestation period is 21 days. The eggs hatches in 21 days. So those are the properties or the characteristics of the domestic fowl. So we'll be going to the next specimen now, which is going to be our specimen C. And our specimen C is, we have our specimen C to be the picture of the uterus. That is what we will be looking at right now. The picture of the uterus. Containing fetus. containing fetus. Yes, that is what we'll be looking at right about. So the picture, this is the picture we're talking about here. We have the picture here. We can see that the picture is here. So this picture is actually carrying a whole lot of information. We're starting with talking about okay, the fetus. Here we can see the fetus showing in this part of the uterus. Now it is the uterus is also called the womb. It is also called the womb. So it is in the uterus that we have the development of the fetus. Now we quickly look at how reproduction occurs in the female reproductive system. Now this particular diagram is not showing the what we call it the ovaries. It's not showing the ovaries on both sides. Here we have the ovary here and we have another ovary here. So the ovaries are responsible for producing what we refer to as the egg cells. So when the egg cells are produced, they come into the fallopian tube here or here, and then they await the arrival of the sperm cell. So the sperm cell comes through the vagina into the cervix, into the uterus, and then it moves into the fallopian tubes, into either of the fallopian tubes. That is when there is no fetus in the womb or in the uterus. So the sperm cell moves through the vagina, the cervix, into the uterus, and it finds its way into the fallopian tube. And when the fallopian tube has mature egg cell, the mature egg cell is fertilized. The mature egg cell is fertilized, and when these mature egg cells, when they are fertilized, it turns into fertilized egg cell. It turns into fertilized egg cell. And when we call the fertilized egg cell, we call it the blastocyte. We call the fertilized egg cell, we call it blastocyte. So the blastocyte move into, from the fallopian tube, it moves into the womb to find a place of attachment to the wall of the womb. Now, when it finds a place of attachment to the wall of the womb, that is when we see that conception has actually taken place. So the diameter of the womb now, if you look at the womb, talking about its longitudinal distance, talking about from the top to the bottom here, if you look at the distance across like this, it has a distance of about 8 centimeter. It has a distance of about 8 centimeter, and across like this, it has a distance of 5 centimeters. Then we have the wall here to be about 4 centimeter thick on both sides. Now, when the cervix opens, this part here called the cervix, it has its own function. The function of the cervix is to permit blood cells out during menstruation. The cervix opens to permit blood cells. Now, what brings about what we refer to as the blood cell? So the inner lining of the womb, we call that part the endometrium. The inner layer of the lining of the womb or the uterus is called the endometrium. So every time there is no fertilization or no fertilization, as the case is in the womb or in the fallopian tube, then the mature egg cell that was not fertilized will be shed off. That is the endometrium, the inner lining of the womb. It will be shed off and it passes through the cervix, and that is what we refer to as the menstrual cycle. That is the shedding of blood or the uterine shedding during the menstrual cycle. Also, we have the uterus permitting sperm cells entrance into the fallopian tube, passing through the uterus. The cervix also opens up, this place here opens up to permit the sperm cell from coming in, going into the fallopian tube through the uterus. So those are the functions of the cervix. So let us quickly see. The uterus is made up of, or it consists 3 layers. We have the perimetrium, that is the outermost protective layer. This part here is called the perimetrium. And then we have the myometrium, the highly muscular middle. This part here, it is very, very muscular, and this is what expands during pregnancy. It is this part of the uterine lining that expands during pregnancy to allow for the accommodation of the fetus, and then it contrasts again during childbirth. It contrasts again during childbirth to help to push the baby out. And then we have talked about the last one, which is the endometrium, the inner lining of the womb, which is where the unfertilized egg cells usually are, and then they are shed off during the menstrual cycle. So the uterus play a key role in the reproductive health, and it functions in three major ways. In pregnancy, the uterus stretches to grow to allow the baby for accommodation. It accommodates the baby and can also contrast to help push the baby out during childbirth. And number two, fertility. The uterus is where the fertilized egg implants during conception. Yes, like I said earlier on, after fertilization has taken place from the fallopian tube, then the fertilized egg cell, or we call it the blastocytes, come into the wall of the uterus to find a place of attachment, and that is where the baby grows. And then the third one is the menstrual cycle, like we have said, that the uterine lining is where the blood and the tissue come from during menstruation. And then lastly, we talk about the four sections, the four major sections of the uterus. The four major sections of the uterus. We have the part called the fundus. The fundus is this part here, the part that connects the fallopian tube to the uterus. That is where we refer to as the fundus. And then we have the corpus. The corpus is the main body of the uterus, this part here, that houses the fertilized egg cell or the blastocyte for it to develop. That is what we refer to as the corpus. And then we have the isthmus. The isthmus is between this place here, between the corpus and the cervix. That is where we have the isthmus. The isthmus is between the corpus. This is this corpus, this part here that houses the fallopian, that houses the fetus. And so between the corpus and the cervix, because this is the cervix region here. So between the corpus and the cervix is where we now have this part here called the isthmus. And lastly, we have the cervix. The cervix is the lowest part of the uterus, and the cervix opens into the vagina. The cervix opens into the vagina. So those are the parts of the of the uterus. So like we have said, there are three major stages rather in the production of organisms. In the production of organisms, particularly in humans, we have it moving from, at the early stage when the egg is being fertilized, it is called the zygote, and that takes about a couple of days from the first minute of fertilization to about a week or thereabout 2 weeks. And from 2 weeks to 8 weeks, it is termed embryo. At the early stage, it was called zygote, and then between 2 weeks to 8 weeks, it is called embryo, and after 8 weeks, it becomes fetus. That is what the growth stages look like. And then the fertilized egg is implanted in the uterus. I've said that earlier on. And then during childbirth, we have something called the involution, which is the shrinking of the uterus to permit childbirth. So those are the things we will be looking at for the uterus and the uterine lining. So let us quickly make a little note for that so we'll be able to get it right. Like I said, the first thing we'll be looking at is the length. I said the length of the uterus is about 8 centimeters, and then we have the width. The width is about 5 centimeters. We have the thickness of the walls. Thickness of walls to be 4 centimeters. We have it to be 4 centimeters. This one here, I said from top to bottom, and this one is from sides, from left to right, so we don't mismatch the dimensions. So I said that the cervix helps to permit blood cells permit blood cells out during menstruation. Also permit sperm cells entrance into the uterus. And then we talked about the four several sections. The four sections. We have the first one to be the fundus. We have the second one to be the corpus. We have the third one to be the isthmus. And we have the fourth one to be the cervix. So the fundus is the part that connects to the fallopian tube. We have the corpus, that is the main body of the uterus, the main body housing the fetus. We have the isthmus to be the part between the corpus and the cervix. And the cervix is the lowest part of the uterus. So we also talked about the endometrium. Endometrium is the inner lining of the uterus that is shed during menstruation. Also, we talked about the myometrium. Yes, the myometrium. The myometrium is the muscular part. It is muscular, enlarging the muscular part enlarging to accommodate the fetus, and then it also contrasts to push out baby during childbirth. Yes, it also push the baby out during childbirth. So those are the things we talked about. We made mention of the functions of the uterus for pregnancy, for fertility, as well as menstruation. So those are the stages. We also said lastly here that we have the stages to be zygote. That's 0 to 2 weeks, to the fetus, to the embryo, which is 2 to 8 weeks, and then to the fetus, which is 8 weeks to 40 weeks, thereabouts. So that is how that is. So like I said, we have the involution. The involution is the shrinking or the contraction of the uterus after birth, and then we have the blastocytes to be the fertilized egg. And gestation period for the specimen C is usually 9 months. It's usually 9 months. That's about we have it to be about 270 days there about. So that is that about specimen C. So we'll be moving to specimen D now. Specimen D is we have our specimen D. Specimen D is the longitudinal section. The longitudinal section of the pride of Barbados plant, the pride of Barbados flower. The pride of Barbados flower, the longitudinal section of that particular plant is what our specimen D is. So we will quickly be taking that now. So what do we have for the ovary of the longitudinal section of the pride of Barbados, longitudinal section of the ovary of the ovary of the pride of Barbados. So lest I forget, we have the specimen C, organism that produce specimen, or the organism that have specimen C. The kind of reproduction that they have there, we call it the viviparous kind of reproduction, viviparous, because they actually have the ability to give birth to their young ones alive. So it is called viviparous. Before we get off that, so that is what that particular organism is, the viviparous organism. So the specimen D that we're talking about now, that is the longitudinal section of the ovary of the pride of Barbados. Let me quickly correct this so we don't get it wrong. The longitudinal section of the ovary of the pride of Barbados. The ovary of the longitudinal section of the pride of Barbados. So we said that specimen D is the longitudinal section of the ovary of the pride of Barbados. Now I will quickly explain specimen D and specimen E together, so we need to be very attentive for this. Now, the characteristics of the pride of Barbados. The characteristics of the pride of Barbados is that the flower, their symmetry. It has bilateral symmetry. Bilateral symmetry. It has bilateral symmetry, and the placentation is marginal. It has marginal placentation. So when we talk about placentation, there are different types of placentation that we have. We have different types of placentation. Example, we have the axial placentation. This one is found in tomato and in orange. We have the marginal placentation, like the one we're talking about now, that is found in the pride of Barbados or the flame of forest. We have the parietal purple, the parietal placentation that is found in purple. So we have the free central also, the free central which has water leaf. It has pepper and pear. The free central that has water leaf as its example, pepper as well as pear. And then the last one, which is the basal. The basal placentation has the sunflower in that category. So we have made mention of 5 placentations now. We talked about axial placentation to have tomato and orange, the marginal placentation majorly for the legumes like the ones we are talking about for the pride of Barbados, as well as flame of the forest. We have the parietal placentation for purple. We have the free central placentation for water leaf, pepper and pear, as well as basal placentation for sunflower. So those are the types of placentation that we have for this particular plant, leading us to this particular types of placentation. So for the specimen we're actually talking about, which is the pride of Barbados, its own placentation is the marginal placentation. Like we can see here, the marginal placentation, that is its own type of placentation. And then the type of ovary that is found there is half inferior ovary. We have three types of ovary as well. Let me quickly put this. Half inferior ovary. So like I just said, we have three types of ovary. We have the half inferior ovary, we have the inferior ovary, as well as the superior ovary. These types of ovary are based on the positions of the floral parts respective to the receptacle of the plant. When you have the receptacle to be lower in position than the floral part of the plant, that is called the inferior ovary. And when the receptacle is carrying the floral part, that is the half inferior ovary, like the one we are talking about now for the Pride of Barbados. It is half inferior ovary. And the scenario where we have the floral part below the ovary, we have the ovary below the receptacle. That is what we refer to as the superior ovary. So we have three types of ovary. Like I've just mentioned, we have the superior ovary, the half inferior ovary, and the inferior ovary. But for this particular specimen we're talking about, its own type of ovary is the perigynous ovary, or the half inferior ovary. And then the reproductive structure, it is bisexual in nature. It is bisexual in nature, or we can also call it the hermaphrodite. That is telling us that it has the ability to reproduce on its own because it possesses the two gametes, the two gender gametes. Those are the gametes that make it possible for reproduction to take place. And then we talk about the biological importance of this particular plant. We have one, the sepal to be brightly colored. Sepal is brightly colored, like we can see in this picture here. You can see the sepal and the petal, that they are brightly colored. See, the sepal is green in color, the petal is red in color. So it is very, very beautiful. It is brightly colored. So that is this is what I'm showing us. This is the petal, the sepal here, the sepal as well as the petal in red color. So we also said that one petal is modified into tube-like structure, or the honey guide. One petal is modified into tube-like structure, or honey guide. And then we have large fan-like brightly colored petal. What we have here, we can see that the petal is large, brightly colored petal, and it has yellow-pink color. This particular part here, it has yellow-pink color. So that is what the petal is. It is large, fan-like. And then it has long, brightly colored filaments. Long, brightly colored filaments, you can see. This is what we refer to as the filament. This particular strand here is what we refer to as the filament carrying the anther. That is, the filament and the anther make up the stamen, like you can see. And then the agent of pollination for this particular substance is insect. It is an insect pollinated flower. Example of the pollinators, we have bee, we have butterfly. So those are the examples. So when we talk about the summary of the floral parts, we have the stamen to be yellow, black, or pink. We have it to be 10 in number, and it is very essential. The carpel is green. It has 1 as its number. The sepal is 5 in number, and it is pink or green in color, as well as the petal, which is pink red, which has a total of 5 in number. So those are the characteristics of or the properties of the Pride of Barbados plant. So this is how the flower looks like, and that is our specimen, our next specimen, Specimen E. You can see this is how its flower looks like, the flower of the Pride of Barbados. You can see the floral part that we just discussed now. This part here is the floral part that we just discussed just now, having the bright colored petal. You can see the bright colored petal having yellow, pink, red color, and then underneath each of them, that is where you have the sepal, which is green in color. you can as well look at this as the filaments carrying the stamen, carrying the anther, forming the stamen. So all of this part here is the leafy part of the pride of Barbados. If you look closely, you see that it has a particular type of seed here. The seed is long and it has its own type of placentation, which is called the marginal placentation. So that is that about the specimen. So let us just quickly round up what we are talking about for this particular specimen. I said one petal is modified into a tube-like structure. One petal is modified into tube-like structure. And then the third one, it said it has large fan-like brightly colored petal. That is usually yellowish pink. Usually yellowish pink in color. And then we have long brightly colored filaments as well. Long brightly colored filaments. And then it has agent of pollination. It is agent pollinated. The agent of pollination is insect. Insect pollinated flowers. It is insect pollinated. Example of the insects, we have bees, we have butterfly. Those are examples of the pollinators. So our next specimen, we have already discussed that. We have shown us the picture of the specimen, that is specimen E, to be the flower of the pride of Barbados. The flower of the pride of Barbados. Sorry, the leaf of the pride of Barbados. So we will not be dwelling much on that because we already showed us the picture. This is the leaf here. We have the leaf here, so that is what the leaf looks like. You can see it's having tiny leaves in between them. So that is that about that. So our next specimen is going to be specimen. I've talked about specimen E now. So our next specimen is going to be specimen F now. So our specimen F, specimen F, which is the Panicum plant. The Panicum plant, or we call the Panicum plant, or we also call it the Guinea grass. The Guinea grass. That is what it is also called, the Guinea grass. So that is our next specimen that we will quickly be looking at. So what is the Guinea grass? The Guinea grass is a forage, is a plant that is mostly eaten by the ruminant animals. It is fed upon by ruminant animals. It is a perennial plant, that is, its life cycle is more than a year. It has more than a year, in fact more than 2 seasons. Its seeds have oval shape and it is wrinkled with light green colors. The lower branches have seeds. They have seed heads arranged in clusters. The lower branches of the plant have seed heads that are arranged in clusters, or they are packed together. And then it has short creeping rhizomes as roots. It has high growth rates. This particular plant has high growth rates. So this particular material has high growth rates. Like I said, it is very, very germinative. That is, it easily grows even in places you least expect it to grow. It sprouts and it germinates there, and it is fed upon by ruminants, organisms that feed on plants, or let me not just call them herbivores, but those are the names, organisms that feed on plants. Also for the production of hay and silage, for production of manure, etc. So we have it called the Panicum plant or the Guinea grass. So I said that it is a perennial plant. It is perennial. That is lasting more than 2 seasons, more than 2 planting seasons. It has that's for the life cycle. That's its life cycle. And then the seeds are oval in shape. The seeds are oval wrinkled. Seeds are oval wrinkled with light green. Then the branches, the stem has lower branches with the seed heads arranged in clusters. Lower branches of the plant. The lower branches of the plant have their seeds, have seed heads arranged in clusters. Or packed together. Also, we have we call them the short creeping rhizomes. The roots are short creepers. Roots are made of short creeping rhizomes. And then they are fed upon, like I said, they are fed upon by ruminants. Let me quickly say this, that they have high growth rates. That is, germinate extensively. Extensive germination. And then we have them fed upon by ruminants. Majorly ruminants like the cattle, the goats, etc. And they are also used for the production of hay. And silage. So those are the functions of the specimen F. So the next specimen we'll be talking about now, that's specimen G, is the Cocoyam plant. Specimen G is the Cocoyam plant. So what is this particular specimen and what is its function or purpose? Specimen G. It is from the family Araceae. Family name. The family name is Araceae. That's the family name. You could be asked for the family name, so that is the family name. It has broad green leaves. They are oftentimes called the elephant ears because of the shape. The elephant ears. Look at what I'm talking about here. You can see the shape of the leaf. You can see how big it is. So it is oftentimes called the elephant ear. That is what it is called. It has or forms edible roots. It forms edible roots. As combs. edible roots as comes, and that is in the case of its own type of root, which is the cocoyam tuber, as well as it being herbaceous. It is also herbaceous. It is herbaceous, so it can be used for local herbs. It is an herbaceous plant that is used in the production of local herbs, used to prepare local herbs. And then we said that it is also a type of perennial crop. It is an example of a perennial crop, telling us that it has longer than two planting seasons. It germinates longer than two planting seasons, greater than two planting seasons. So that is what the specimen G is about, and we can see the picture right here. So for that particular specimen, this is what its seed looks like. This is the seed of, the storage fruits, or the storage food of that particular specimen G, which is the cocoyam plant. So this is going to be our specimen H. Specimen H is the yam, we call it the yam, or the comb of cocoyam. Specimen H is the comb of cocoyam. So it can be called the taro comb. The taro comb. So it is edible in nature. It matures in 8 months. Like we have said earlier on, it is edible in nature because it is highly rich in carbohydrates. It is edible roots of the cocoyam plant. The edible roots of the cocoyam plant. That is what this particular specimen eats, and it matures in 8 months. Matures in 8 months. The tuber is white. The tuber is usually white. The tuber is usually white when peeled. Usually white when peeled, turns pink. And then the leaf also, I've said that earlier on, that the leaf of the plant is used for herbs. So this is the specimen we are talking about here. This is the particular specimen we are talking about. Our next specimen is specimen J. Specimen J, which is the dry humus. Specimen J is our next specimen, and it is the dry humus. Dry humus. So when we talk about the dry humus, this is its picture here. You can see how it looks. It has dark organic materials. That's dark organic material that is made from fossils. Fossils that we call them fossils because they are made from remains of plants and animals, dead or decaying remains of plants and animals. And it contains nutrients. They are reduced in moisture content or water level. It has limited moisture. In fact, sometimes it is absolutely dry. It has no water content or moisture. And then it flakes or dusts when they are being touched. It flakes or dusts into grains when they are touched, and it provides nutrients to plants. But because it is dry, it will not be able to give as much nutrients to the plant as possible, because the plant cannot absorb nutrients that are in this particular soil in the raw form. It has to be dissolved in the soil for the plants to be able to take them in. So that is it. We said that they have dark organic matter, dark organic material formed from fossils, remains of plants and animals. Also, that it contains nutrients. Contains nutrients, and it has reduced moisture. It has reduced moisture or water content. It flakes or dusts into grains. It flakes or dusts into grains when touched, and then provides nutrients to plants. Provides nutrients to plants. So these are the characteristics of the particular soil. So don't forget, like I said, that because it is dry humus, it has the nutrient in it, but it will not be able to give it to the plant because the plant cannot absorb the nutrients in the solid form. The nutrient has to be dissolved first. The nutrients have to be dissolved in water, then it is this water that the plant will be able to absorb into their system. So our next specimen, the next specimen we shall be looking at right now, is going to be Specimen K. Specimen K. Specimen K, which is talking about moist or wet humus. Moist or wet humus. That is going to be the next specimen. Now, it is just something similar to the dry specimen that we have talked about. The only difference in them is the fact that this one is wet, and because it is wet, it has the ability to allow the plants to absorb the nutrients that is in it. That is the major difference. Allows plants to absorb nutrients found in it. So let's quickly say for this one, the previous one provides nutrients which is not absorbable. To the plants because the nutrients need to be dissolved first. Before they can be absorbed. So it is not that it doesn't have the nutrient, it has the nutrient, but because it does not have moisture in it, the nutrients will not be able to move into the plant. Also, let me quickly add is that it has no living organism. There are no microbes. Absence of microbes in this particular part. Absence of microbes. So these are the major differences between the specimen J, and which is the dry humus, and the specimen K, which is the moist humus. In the moist humus, there is we have the presence of humus. The presence of microbes. Microbes can be found in the wet humus. So those are the major differences between the dry humus and the wet humus. And so we move to the next specimen, which is Specimen L. Specimen L. So our Specimen L is the ripe orange fruit. That is our Specimen L. It is a citrus. Ripe orange fruit is a citrus. The fruit, this is how it is being descended. It is descended from being fruit to true fruits. True fruits, which can be true fruits from ovaries, formed from ovary. So we have the simple fruit. It is also called simple fruit. Simple fruits or a monocarpous flower. To the succulent fruits, succulent fruits, succulent fruits as well. To we can have the Hesperidium. The Hesperidium. We can have the Hesperidium, and that is where we now have the class, e.g. orange. So this is how it is being descended from fruits to true fruits to simple fruits to succulent fruits, then the Hesperidium. It has waxy skin. It possesses waxy skin. Yes, you notice that the skin is oily, and then the placentation is axial. Placentation is axial. Remember we talked about placentation earlier on. Placentation in the orange is axial. You have tomato in that particular class as well. And then it is spherical with outer peel. It is spherical in shape. Spherical in shape with outer peel. Outer peel called the epicap. The outer peel is called the epicap with underlining fibrous layer. Underlining fibrous layer. The underlining fibrous layer, which is called the mesocap. Which is called the mesocap. So the underlining fibrous layer is called the mesocap, and then we have the tiny part. That is the juicy part of the fruit. The juicy part is called the endocap. Juicy part is the endocap. So the outer part or the outer layer, the peel, is called the epicap. We call it the epicap. We have the mesocap, which is going to be the fibrous lining, and then we have the endocap, which is the juicy part of the plant. This is an example of what we are talking about here. You can see the orange. So this outer part is called the epicap. This part here is called the epicap. Then this part here, this part in the middle that has tiny white here, is called the mesocap, and here is where we have as the endocap. Are we together? And then we have it. We say that the plant undergoes asexual reproduction. The plant undergoes asexual reproduction. Undergoes asexual reproduction. It undergoes asexual reproduction, and then the seeds are naked. Sorry, seeds are not naked. The seeds are not. The seeds are not naked. That means they are hidden. That is, they are hidden. The seeds are hidden in the fruit. So that is what we have for the specimen. That is what we have for the specimen. So this is what we have for the specimen of the ripe orange fruit. So our next specimen is going to be specimen. We have the next specimen, specimen M. Sorry, specimen M. Specimen M, which is the longitudinal section of the coconut fruits. Longitudinal section of the coconut fruit. So this is what we are talking about. This is how the longitudinal section is when it is cut from top to bottom. So we have this as its labeling. So we have the exocap, the mesocap, the endocap, as well as the endocap. Are we together? So we said that the water dispersed, okay, it is a water dispersed fruit. Specimen M is a water dispersed fruit. Water dispersed fruit. It's a water dispersed fruit with leathery epicap. With leathery epicap, we call it leathery epicap. Leathery or hairy epicap. Yes. So it has round elongated fruits. It has round elongated fruits. Round elongated fruit. So round elongated fruits. It also has white fibrous oily meat. White fibrous oily. It has white fibrous oily meat. The oily meat, that is the part that we eat. This part here, this part inside here is the white fibrous oily meat. That is the part that we refer to as the endosperm or the endocap. So we have that, and it contains 51%. It contains 51% or 51.7% kernel. Contains 51.7% kernel, and then 9.8% of water, as well as 38.5% of shell. So that is the constituent of the coconut plant. 51.7% kernel, 9.8% water, and 38.5% shell. The oil is rich in protein. It has 3 layers or 3 pores. It has 3 pores. The oil is rich in proteins. So the fruit now, the fruit of the coconut, usually we know it has 3 eyes. Particularly when we have removed this outer exocap, when we have removed this exocap, it usually has 3 nodes, 3 eyes, and so those 3, 2 of them are actually woody, but the last one is the one that we can pierce to have way into the particular part of the organism. So on the micropyle here is what I'm talking about. Somewhere around here we have 3 holes. 2 of them are called the plugged pore, while the last one, which is possible to pierce through, is called the functional pore. So 3 pores. It has 3 pores. That is 2 plugged pore, 2 plugged pores, and 1 functional pore. 1 functional pore. So those are the pores that we have in the coconut plant. So let us quickly look at its functions now. Its function: the oil is rich in protein. Oil is rich in protein. That is when it is being consumed, and vitamins as well. Protein and vitamins. So when it is used for cooking, it has these particular properties. And then it is also used for skin care. Also for skincare products. It is found in majority of the skincare products. That is that. Also, we have the tree itself to be used. The tree is used for wood for furniture and some other things. Also, it can be used for shades and for shades. When we cut the branches of the tree, it can be woven together to form shades. Also, the hairy shell is combustible. Hairy shell is combustible. That means you can use it to make or ignite fire. So those are the properties and the uses of specimen M. And the last specimen we have is specimen specimen N, which is the longitudinal section of fresh chili pepper, fruits. That is the last specimen that we have for this segment. So actually, this is how the fruit looks like. This is the fruit itself. But when we talk about its cross-sectional area, when we talk about the longitudinal section of the fresh chili pepper fruit, this is what we will be having. When we talk about the longitudinal section, this is what we will be having from the previous picture that I showed us. So it is a berry fruit which is usually hot and peppery. It is a berry fruit which is usually hot and peppery. The seed is attached to the wall of the placenta, so it also has marginal type of placentation. Then it is hidden. The seeds are hidden, so they are not naked seeds. The seeds are not naked, so they are hidden. And then the perfect flower. It is a perfect flower containing stamen, and it is capable of self-pollination. It contains stamen, that is the pistil and the carpel. So we look at the characteristics now. Let us quickly write out the characteristics that we have for this particular specimen. The characteristics now. So the first one, I said it is a berry fruit. A berry fruit. A berry fruit that is hot and peppery. It is hot and peppery. The seeds are attached to the wall of placenta. Seeds are attached to the wall of placenta. Yes, the wall of the placenta, which is free central. Free central. That is its own type of placentation. And then we said that it is it has no naked seeds. Absence of naked seeds. That means the seeds are hidden. And lastly, that the flower part it is a perfect flower. We say it is a perfect flower. Containing stamen, and that it can self-pollinate, capable of self-pollination. So this is what we are actually talking about here, this particular plant. You can see that all of the specimen, all of the seeds here for the placentation, all of the seeds, they are found on the particular placenta which is found in the center of the plant. That is why it is called free central. So this is what it looks like. So those are the specimens that we have for the WAEC practical for 2023. We have listed them. I'll advise every one of us to pick this, listen to them, try to understand them, and if you have any question arising from this particular tutorial, you can drop your question at the comment section. Please share, like, and subscribe to this channel. Thank you very much, and God bless.